Okay. Okay. Welcome, everybody. Thank you for being here. This is going to be a fantastic presentation, I know. And for those of you who may be guests, we are the Society for Veterinary Medical Ethics. Please look at our website that is down there below. And if you have any questions, feel free to email at info. And let me tell you a little bit about who we are. A community of people who really love ethics and recognize how important it is for the veterinary profession, I think now more than ever. And we promote discussions, debates. We support each other, talk about things that are relevant to veterinary practice. And one of the main missions is to educate and do research. And so today is going to be a fantastic opportunity to add to that great education. And if you do join, you have access to exclusive content. We have these wonderful monthly meetings, and thanks to Barry Kipperman for really putting together just an incredible list of speakers. You can become a leader, you can mentor. We have student groups, which is fantastic. So you have some wonderful opportunities. And we're not exclusively veterinarians. We have attorneys, we have MDs, PhDs. So anybody that is interested in ethics in the animal world are welcome to join us. So again, networking opportunities, that's really one of the reasons I joined. Is I came to some of the meetings and discovered that I loved the people that were there and wanted to be a part of it. There are discounts too. We have an outstanding basic veterinary ethics course, which is complete now and getting updated, which is fantastic. So you have a discount for that. And then if you're a speaker, we have honorariums and we give stipends to students. We do a student competition on an ethical issue every year. So again, please look at the website. If consider joining us, we'd love to have you. So let me go ahead. I want to introduce our speaker. And this is Dr. Andrew Knight, who is originally from Australia. I think you'll figure that one out. And ever since helping launch the campaign against the live sheep trade to the Middle East in the 90s, so he's been doing this a while, he's advocated on behalf of animals. And so for nearly a decade prior to 2012, he practiced veterinary medicine, mostly around London. And then in 2013 to 14, he directed the Clinical Skills Laboratory and taught animal ethics, welfare, veterinary practice management, surgical and medical skills at Ross. So that's very impressive. He's currently an adjunct veterinary professor of animal welfare, affiliated with the School of Veterinary Medicine, College of Environmental and Life Sciences, Murdoch University in Western Australia, and the School of Environment and Science at Griffith University, Queensland. He's also affiliated with the University of Winchester, where he established and directed its Center for Animal Welfare from 2015 to 2023. And this center grew to become one of Winchester's largest and most active research and knowledge exchange centers. He's also an EBVS European and RCVS veterinary specialist in animal welfare science, ethics and law, American and New Zealand veterinary specialist in animal welfare, fellow of the Royal College of Veterinary Surgeons, and principal fellow of Advanced HE. His many books include the Routledge Handbook of Animal Welfare, The Costs and Benefits of Animal Experiments. He has approximately 150 and 80 popular publications, an extensive series of social media videos on vegan companion animal diets, which is very fascinating, climate change and the livestock sector, invasive animal research, educational animal use, humane clinical and surgical skills training, and other animal welfare issues. Several have been published in leading scientific and medical journals, such as New Scientist and the British Medical Journal USA and PLOS One. He's delivered around 250 presentations at conferences and universities internationally and has organized or chaired 7 conferences and seminars. He regularly works with animal welfare charities to advocate for animals and is frequently interviewed by the media. You can see why. He has been honored with 15 awards and 22 research grants, including the Society for Veterinary Medical Ethics Schomer Award, which is something that we give, and the Humane Society Veterinary Medical Association Humane Achievement Award, both in 2019 and both well-deserved. He enjoys teaching and was delighted to receive a university student-led teaching award in 2017. Since 2019, he's secured vast amounts of funding, 95% of which is external, which is important these days. In addition to all of that, he'd like us to know that he's also a keen outdoor enthusiast. So 5 minutes a week, he gets to go outside and see what's going on. So, okay, yeah. So Andrew, thank you again for being here. And please go ahead, screen share, and we look forward to you speaking with us.
Thank you so much, Sue, for that very kind introduction and also for this kind of opportunity to speak here. I would add that my outdoor exploits have far greater enthusiasm than skill, which can occasionally be hazardous in places like the mountains. Anyway, I'm not here to talk about that, sadly, although I would very much like to do so. Here to talk about educational animal use and alternatives. And my foray into this field really began when I was a veterinary student at West Australia's Murdoch University, what seems an awful long time ago. And you can see the main campus there. It's very beautiful. We have kangaroos and wallabies hopping around the forest down behind the veterinary school. We had bunny rabbits roaming around the lawns there early in the morning. It's a very beautiful campus in Western Australia. I entered veterinary school as a student in the late 1990s with the vague idea that harmful animal use was probably required in parts of the curriculum, and also the vague idea that humane teaching methods were probably available. But I was ignorant of what the details of either of those might be. And I rationalized to myself that if I was required to harm animals in the curriculum, it would be worth it because I'd be able to do so much more good for animals after graduation as a qualified veterinarian. So thus unprepared, I entered first year of the veterinary course here, and I found in the first year classes that we would dissect animals such as rats, snails, slugs, earthworms, lots of body parts for abattoirs, and for reasons that always seem mysterious to us, there seemed to be a strange obsession with dissecting lampreys. Most of these animals appeared pinned out on wax dissecting trays, looking very different from the living, breathing animals that they had once been. And this helped me to dissociate from them as living, breathing animals that were being killed for our teaching. This facilitated my ability to avoid thinking seriously about this issue. And this strategy of not really thinking very much served me very well until the end of first year. And at the end of the first year, something changed. There was a particular laboratory class, which was a cell biology laboratory class. And during this particular lab class, the animals were not appearing pinned out on dissecting trays ready for us to dissect. This time they were in fact the living, breathing animals that were running around in cages shortly before the start of the class. When they were killed by demonstrators, so that we could then go in and dissect out the intestinal tracts with some of the cells still, very fresh and somewhat alive, to measure the uptake of glucose in various solutions by the living intestinal cells. And no doubt to demonstrate some sort of biological principle that had been established decades ago and could easily have been learnt through videos or books. I was sure that alternatives must exist to this kind of alternative, even though I didn't know exactly what they might be. And so I went to the demonstrators in charge of this class, and I told them that I didn't want to participate and requested a humane alternative. Unfortunately, on this occasion, I was poorly prepared. I only voiced my objection on the morning of the laboratory class. I didn't know what the alternatives might be or the other courses around the world where they were successfully used. My request was denied, with the result that I boycotted this class, which ended up costing me a grade in that particular module. However, this ended up saving the lives of about, sorry, I should say this stirred up a great deal of controversy with some students and some faculty members supporting me, others opposed. I was called into the office of the people in charge of the course, who spent an hour and a half trying to interrogate me and to change my views. They gave me dire warnings that this cell biology lab class was only the tip of the iceberg compared to what I'd have to do to animals later in the vet course, and suggested that I reconsider my choice of career. This wasn't successful in making me drop out of the veterinary course, as seemed to be the intention. But it did motivate me to go away and learn properly about what the humane teaching methods were that were available for these sorts of labs, which courses around the world they were being successfully used in, and what the evidence was from educational studies of student learning outcomes about the efficacy of these sorts of methods compared to traditional harmful animal use. So I'm going to share with you some of what I learned. I would say that the amount of controversy that my boycotting of this one laboratory class caused resulted in the lab actually being dropped from the curriculum the following year, ostensibly just for financial reasons. And I was delighted that at the cost of just losing a grade in that particular module, we had saved 30 to 50 rats from being killed every year. And in the years and decades that have passed, that's an awful lot of animals. So it was well worth losing a grade in that module, I would say. Harmful animal use within veterinary curricula and other life and health sciences education tends to fall within two broad groups. There's animal use in preclinical subjects such as anatomy, physiology, biochemistry, pharmacology, and so on. And I expect that most people attending this presentation will have gone through such education, be well aware of these sorts of animal uses, and have seen perhaps pictures like these. And apologies for anyone that wasn't aware. I should perhaps have said that prior to showing these images. The other type of harmful animal use, I should say, is veterinary clinical and surgical skills training, which occurs obviously at the higher years of veterinary courses. So with respect to the preclinical training, there's commonly use of purpose-killed animals within anatomy dissection classes. The practice at my veterinary school, which was common in veterinary schools around the world, was that greyhounds would be donated for teaching purposes once they were no longer racing fast enough to be profitable on the racetrack. And they would initially be used as blood donors in a blood donor colony. And they would also then be killed for use in anatomy dissection. And a group of around about 4 students would be given a greyhound at the start of semester, and each week we would dissect a different part, and it might be a foreleg or a hind leg or a chest cavity, a thoracic cavity, and so on. And in between, the greyhounds would go into the cool room. So we had a lot of dissection of these greyhounds in our anatomy class, along with various other body parts as well being used within student dissection projects. And this will be familiar to those of you who've gone through veterinary training and perhaps some other life sciences education at university level. There is also the harmful use of animals in demonstration laboratories, such as physiology, biochemistry, pharmacology, to demonstrate the effects on animals of things like drugs, parasites, and in our physiology course, it was also things like forcing the animals to breathe different gases, occluding the air supply entirely, dissecting into and ligating nerves and arteries and things like that to observe the effects on animal physiology. Injecting them with various drugs to do things like monitor dropping of the heart rate, for example. At the end of these procedures in our physiology labs, during which typically student groups would anesthetize sheep prior to the start of the procedure, At the end of these procedures, any surviving animals would then be killed by anesthetic overdose before regaining consciousness. And then at the clinical and surgical level, there would be typically terminal surgery labs in which healthy animals are used as teaching models, with student groups practicing surgical procedures on them and then killing them under anaesthesia prior to recovery. In our veterinary course at Murdoch University, we would use young pigs. There was also some limited use of greyhounds from the teaching colony, and sometimes sheep as well, but primarily pigs were used. So this sort of picture is common in veterinary schools, or was common in veterinary schools across Australia, North America, and indeed virtually all regions of the world in which there is westernized veterinary surgical training, except the United Kingdom, where interestingly harmful animal usage at the surgical and clinical level was eliminated decades ago and replaced entirely with externships and surgical training models and mannequins. And I'll talk more about those later on. Because the teaching laboratories, in particular things like physiology, demonstration experiments and biochemistry experiments, tend to be a standard set of laboratories repeated in courses around the world. There have also been developed a standard set of alternatives to replace animal use within these laboratories. And the alternatives tend to be things like these: high quality videos and computer simulations of animal dissections or experiments. Students' dissections of cadavers obtained from animals that have died naturally or in accidents or been euthanized for genuine medical reasons or severe and intractable behavioral reasons. Those are ethically sourced cadavers. Interestingly, the term ethically sourced is often co-opted to seek to justify the killing of animals from less or non-ethical sources, as, by the way, is the word euthanasia, which by definition means killing which is not only humanely conducted, but also genuinely in the animal's best interests, which occurs if it's suffering from a severe and intractable illness and prognosis for recovery is poor. Both these terms, ethically sourced cadavers and euthanasia, are often co-opted in an attempt to legitimize killing which is not truly euthanasia and which is not truly ethical. So obviously watch out for the misuse of those terms. Specimens, whether from non-ethical or ethical sources, body parts from animals can be permanently preserved in a variety of ways, thereby at least eliminating the need for animals to be killed year after year. Non-invasive self-experimentation on oneself and one's classmates in order to demonstrate, for example, physiological principles such as increase in heart rate and respiration associated with exercise are used in medical courses such as that of the University of Adelaide and many others around the world. And then we have clinical and surgical skills models, mannequins and simulators, and I'll show some of those later on. And of course, supervise clinical and surgical experiences through internships and externships within veterinary teaching hospitals and associated with veterinary practices and animal shelters are really important as well. And of course, this is the standard way that human surgeons are trained and human clinicians. It's not via practicing harmful procedures upon people, or homeless people. It's which is what we do to homeless dogs sourced from animal shelters. Sometimes it's supervised involvement in beneficial surgical and clinical procedures with real patients. These are images of computer simulations of animal dissections. You will see that there can be high quality images of professionally performed dissections or prosections. Any of you that will recall dissections at university will probably be familiar with our experience, which is that the organs of animals like rats, the abdominal organs, are often small and fragile and friable and quickly destroyed by the clumsy dissections of novice students. And of course, once they've all been mashed up, you can't put them back together. You can much more readily identify and learn about the different organs using image from a professionally performed dissection, such as we see on the left-hand side. In these simulations, the computer simulations, they often include histological microscopic anatomical slides juxtaposed with the gross or macroscopic anatomy as well, which is not something that you get from just performing a traditional dissection on an animal on a dissecting table. Sometimes these computer simulations of dissections can be made available via the internet. This is one that's a very good one of the dog that was produced by Colorado State Veterinary School. And it was freely available and now is available for a fairly small fee for students at veterinary schools around the world. It allows students to focus on different parts of the dog and to study aspects such as the radiographic anatomy, the soft tissue anatomy, bony anatomy, clinical anatomy. If we look at the soft tissue anatomy of the head, for example, you can look at different parts. You can click on different structures and obtain standardized information such as the anatomical origin, insertion, innervation, and action of this masseter muscle here. You can zoom in for a closer look. You can look at the head from different angles. You can rotate the head smoothly, not in jerky shots like you're seeing here, but actually smoothly by dragging a slider button across the screen. You can look at radiographically significant parts such as frontal sinuses here, the lingual veins, the glottis and epiglottis to see where students need to learn where to insert the endotracheal tube when connecting animals to gas anesthetic machines. And some of these simulations, of course, also exist for medical students. This is one called FACE, created by medical illustrator Roy Schneider at the University of Ohio. And in this particular case, students can again drag a button across the screen, a slider button, and in doing so, melt the tissues away, starting at the most superficial, going all the way down to the skull, and then by dragging the slider button in the opposite direction, all the tissues can reappear again. And you can do this as many times as you want, which of course is entirely different from a genuine dissection when you can remove the tissues once, and once that's done, you can't put them back again. There are certain structures that students can click upon, such as the muscle to the side of the nose there, which will then actually display their function. If a student does this in the actual simulation, that muscle will contract, causing the nose to twitch. So the student can see the action that the muscle performs. And there's another one involving causing the eye to blink. So again, this provides functionality which is superior to that of a traditional dissection on a dissecting table. If a medical student sees a head starting to blink or twitch at them, then they need to perhaps be seriously wondering about what they had for dinner last night. This is a digital frog simulation targeted at high school students and junior biology students, and it's a very popular one. Students have to dissect dissecting instruments from a virtual toolkit, drag them across the correct parts of the frog's body, and if so, then they'll see a video run of an actual dissection occurring. So sometimes these simulations have footage within them that might not have been ethically sourced, and the same is true of footage of animals undergoing responses to test drugs, for example. In pharmacology simulations, but at least once that footage has been sourced, it can then be used indefinitely with no further harm to animals. There can be functional diagrams of functional anatomy included, and if you click upon the right button, you'll see blood actually flow through the relevant chambers of the heart. You see arterial and venous blood flow, and again, not something that you can see with a traditional dissection. And you can even learn about the natural history of the frog. If you click upon the right button, you'll even hear what the frog sounds like in its natural environment. And I've tried to simulate this myself when speaking to large audiences, but it's usually come out badly. So I'm too frightened to try to do this to yourselves today, which you can probably be relieved about. Cadavers can be sourced ethically, ideally, or not, and either way, permanently preserved using a variety of different ways. And most veterinary schools will have an anatomy museum, and many of you will have seen these in your own veterinary schools, which are full of such permanently preserved specimens. That's me as a student on the left in our anatomy museum with this sort of sectioned horse's head, and these potted specimens are preserved in formaldehyde or other powerful preservatives designed to prevent dissolution and bacterial putrefaction, along with other chemicals designed to preserve colour. There can be coloured casts of airways and blood vessels whereby animals are heavily anaesthetised, and prior to being killed under anaesthesia, their major blood vessels can be perfused with coloured dyes, and the resultant organs are then, the soft tissues are then eaten away with mild acids over a period of several months, leaving these coloured casts of airways or blood vessels. And that's the kidney of a cow that you can see there in the image. Plastinated specimens, such as the plastinated squid at the bottom of the slide, are produced using several chemical steps and a vacuum in which the fats and the water within these specimens are replaced by polymers, producing a plasticky texture, a faint smell, and permanently preserving these specimens. Tufts University Veterinary School in the US went to entirely the use of ethically sourced cadavers to source all of its cadavers for its anatomy program and indeed its clinical skills training program as well, at least as far back as 2011, if not earlier. This photo is from them. This is a vascular cast of the liver and gallbladder of a dog from an ethically sourced cadaver. And here's one showing the major and minor airways and bronchioli and so on from the lungs of a dog. This is Gunther von Hagen, the anatomist who invented the plastination procedure, demonstrating that even the largest of animals can be successfully plastinated. And last I checked, he had 4 travelling exhibitions of plastinated animals and 1 travelling exhibition of plastinated humans travelling around the world. And if anyone hasn't had the opportunity to see one of his exhibitions, I do highly recommend them. I saw one once at London's Natural History Museum, and it was fascinating to actually see all the muscular and other structures within a variety of animals, with the educational plaques next to each of these describing the functional anatomy of the various structures that were clearly on display. And I was very pleased to see the great big plaque at the front. I'm sorry, what was that? I'm just going to say I was very pleased to see the great big plaque at the front of the display, clearly stating that none of these animals had been killed for the purposes of the exhibition. They had all been ethically sourced, which was great to see. Ethically sourced cadavers can, of course, also be used for teaching clinical procedures. This is Dr. Briony Dixon, who graduated from the University of Queensland Veterinary School in 2011 as the first student to graduate without harming animals in clinical and surgical training at Queensland. Here she is practicing placing a chest drain into the chest of a dog cadaver, and that's an ethically sourced cadaver. There have been many computer simulations of animal experiments in subjects like physiology, biochemistry, pharmacology, and so on. And the early experiments were understandably criticized for being overly simplistic. But modern animal experiments have high quality video clips of responses of animals to actual procedures. They have virtual benchtops using virtual oscilloscopes and instrumentation that students have to set up. On the bottom right, you can see some electrodes. When I did our physiology program, there was a frog sciatic nerve experiment whereby students had to dissect the major muscles of the hind leg of a frog and apply an action potential to the sciatic nerve, thereby witnessing the muscle hopefully contract. This was a very prolonged procedure. It took several hours. It was difficult to set up properly. Students often accidentally applied excessively strong currents to the nerves of the frog in the process of setting up their equipment, thereby rendering the specimen useless for the rest of the experiment. And the experiments were often unsuccessful. Here, students are able to actually apply a virtual frog's hind leg to the virtual electrodes in the bottom right and apply different action potentials to them. And if they get the experiment wrong, it's easy to start afresh and to try again until they actually get usable results, all of course very quickly without harming any animals in the process. And of course, saving significant amounts of money as well, as we'll come to later on. Models have also been developed for high school biology students and also for people like laboratory technicians. The tail vein of the rat is the major vein used to take blood from by laboratory technicians, and this is a model that enables them to practice that procedure as well. For people like veterinary students and animal carers, there are models like these. This is Critical Care Jerry. He is a canine mannequin. And you can practice listening to both normal and a range of pathological breath sounds and heart sounds, feeling for a pulse, practicing mouth-to-snout resuscitation, and so on. These models are often not entirely realistic, but they do provide a great starting point, a bridge between theory and practicing these procedures on living animals and patients. Without these models, students often go straight from theory to a living animal or patient, and that's really not where students should be practicing these procedures for the first time. Here is Dr. Dixon practicing cardiopulmonary resuscitation using a Jerry mannequin. By contrast, the way that students at University of Sydney learnt this procedure up until 2000, which was also common in other veterinary schools around the world, is that dogs would have cardiac arrest artificially induced, and then students would have to attempt to resuscitate these dogs. And if they didn't succeed, then of course the dogs would die. So this is, of course, grossly unethical, but was nevertheless the norm in University of Sydney and many other veterinary schools around the world. So here she is practicing this procedure on a mannequin instead, as many times as she wants and in different ways and under different conditions if she chooses or the instructor chooses. Haptic simulators apply anatomically appropriate pressure to a student's fingertips, depending on where they are inside the simulation. Now, inside the back end of a cow, the plastic hindquarters of a cow, there is essentially a thimble connected to a robotic arm connected to a computer, which is running a simulation. And there is Dr. Dixon practicing pregnancy diagnosis in this simulated dairy cow. The simulation can be programmed to be non-pregnant, late-stage pregnant, other stages of pregnancy, and to have pathological conditions as well. This was developed by Professor Sarah Bailey at the Royal Veterinary College just outside of London, who also developed models for palpation of horses that might have colic. It's an equine colic simulator. And also feline abdominal palpation, which is something that small animal veterinarians routinely do in clinics. Those sorts of models can be very expensive. These beautiful wooden carvings were made by local artists in the Philippines, much more affordably. They're, of course, not soft and flexible, but they nevertheless just allow demonstration of things like how to give pills, how to restrain animals. So a limited number of procedures can still be practiced using very simplistic methods such as these, much more affordably. Alternative veterinary surgical training doesn't involve practicing surgical procedures on healthy animals and then killing them under anesthesia. It ideally involves these three steps. Students firstly using knot tying boards, plastic organs, and similar models to develop their basic psychomotor skills, their suturing and instrument handling skills. Secondly, progressing to simulating the entire procedure on an ethically sourced cadaver. And then the most important part, of course, is observing, assisting with, and then performing under close one-to-one supervision beneficial surgery on real patients, for example, as part of animal shelter neutering programs. This is an example of a surgical simulator. It stands for Dog Abdominal Surrogate for Instructional Exercises, or the DAISY. And it's essentially just a foam cylinder with multiple layers with coloured red threads running around the inside to simulate blood vessels. And indeed, this one has some foam intestines in it as well. When I was a veterinary student, we didn't have any simulators like this, so I simply bought my own from Canada. It cost about $15 and it can be used up to 6 times by simply rotating it. And I took it into a surgical laboratory and I started by practicing my extensive scrubbing for 10 minutes in the right way, gowning, gloving, draping my DAISY, preparing my surgical instruments, blunt dissecting through the various tissue layers, encountering my red thread blood vessels, clamping, ligating those, locating my foam intestines, exteriorizing those, wetting them down with saline so they wouldn't dry out, removing a supposed piece of non-viable intestine, anastomosing the ends together, pressure testing it for leaks, returning it to the abdomen, closing up the various tissue layers one by one. So that took me a good couple of hours as a student surgeon. And of course, this is not entirely realistic, but it's also a very good simulation that's incredibly useful for practicing all those sort of basic skills and steps, and is very cheap. And there's absolutely no excuse, I think, for students practicing these sorts of procedures on real animals for the first time when alternatives like this are readily available. Many other surgical simulators have been developed for veterinary students and also medical students, various soft tissue organ models, bones of various kinds, even osteoporotic bones can be purchased with increased fragility as well. People often raise the point that alternatives don't bleed, so it's they're not amenable to practicing locating the source of hemorrhage and staunching blood flow and dealing with bleeding during surgery. That actually is addressed by the POP trainer, the pulsating organ perfusion trainer in the bottom left. It's essentially a big, large plastic tub into which you can insert major organs, either sourced from an abattoir or ethically sourced, connect up the major blood vessels to a closed circuit containing fluid with some colorant to simulate blood, and a pulsatile pump, which pumps fluid around the circuit. And you can then practice either conventional or minimally invasive surgical on these major organs. And if you nick a blood vessel, they will bleed. And you do have the opportunity to then practice swapping your surgical field, locating the bleeder, clamping it, ligating it, and carrying on. Of course, the best form of alternative surgical training is actually assisting with beneficial surgery on real patients as human surgeons are taught. And that's what's going on in the photo in the top right, which I took when I was a first-year veterinary student. So I'm behind the camera looking at a fourth-year student. That's a final year student on the left-hand side, assisting the surgeon on the right. Here's another example of a more sophisticated haptic simulator. So in this case, the simulation involves minimally invasive surgery, and the surgeon is manipulating the arms, the robotic arms that are performing, in this case, simulated cardiac surgery. And there is only simulation occurring, but there could also, technically speaking, be a patient on the table behind them, or even more scarily, on an operating table on the far side of the world if everyone wanted to trust the life of the patient to the adequate internet connectivity of a situation like that, which is a whole another topic for discussion, a topic potentially of tele-veterinary medicine and telesurgery, remote surgery. Maybe that's something for the future. This is an example of the sort of alternative veterinary surgical program that we established as veterinary students when I was a student at Murdoch University Veterinary School in Western Australia in 2000. We didn't have any highly advanced haptic surgical simulators as like the one I just showed you. What actually occurred was that after going through second year of my veterinary course, we were allowed to have alternatives in biochemistry, but not in physiology. The program leader in charge of physiology was highly committed to invasive animal experimentation and not interested in humane alternatives, even in teaching, and not interested in learning about those or about the educational evidence in this field, and simply denied our request to use them. The result in this case was that after exhausting all avenues available within the university at the time, so going up to the dean of the veterinary school and so on, I had to go outside the university. So I went to the state authority responsible for anti-discrimination legislation in Western Australia and said that I was being discriminated against in my education on the basis of my conscientiously held beliefs against harming animals unnecessarily in my curriculum. They said, oh, that's very interesting. We haven't had a case like this before, but you've made a case that this could potentially violate anti-discrimination legislation. So we'll take this on and investigate this. Concurrently, I also went to the press. And pretty shortly, we had television cameras coming onto campus, and I was giving interviews that made the front pages of newspapers across Australia. And describing a student that wanted to go in to become a healer of animals, being forced to kill animals in their curriculum, even though alternatives existed elsewhere and were well established, and educational evidence indicated that students using them did well. This was naturally a public relations disaster for the veterinary school. So they responded very quickly. They established a committee to examine the entire issue of student conscientious objection. That quickly concluded that the university's values were to uphold people of a variety of religious beliefs, cultural backgrounds, and the students should not be unduly discriminating against people who wanted, for example, not to kill animals in their education. The university committed to making reasonable attempts to accommodate such students by providing alternatives of comparable difficulty. This wasn't limited just to veterinary students. This also applied, for example, to students that in our geology programs that had to go into the Australian outback to mining camps, where they would work sort of 2 weeks on, 1 week off. And if a student had a religious day, a holy day, and that prevented them from working during these sustained working periods, an alternative had to be found for them. So we established as a university a conscientious objection program whereby we agreed to provide alternatives of comparable difficulty to our students. And this was soon adopted by other universities across Australia, New Zealand, and even some in the US as well. So after that, things went fairly quiet for me up until surgical year, the fourth year of my 5-year program. And in the surgical year, we didn't get the same response that we had to in physiology in second year. This time, we didn't have an academic that was flatly opposed and said, no, I'm not going to allow you to have alternatives. This time we had academics who were flatly opposed, but said, well, the university has required us to allow alternatives, therefore we're going to allow it. But because we don't agree with you, we're not going to teach you, but we will examine you. We require you to go and arrange your own surgical training elsewhere and then come back and show us that you can do surgery to our standards. And if you can't do that, we'll fail you. So we said, fantastic, this is a major step forward. Previously, these academics would not have allowed this at all. So myself and one other student, then went out to external veterinary clinics and animal shelters and arranged our own clinical and surgical training out there, which involved assisting with beneficial surgeries with real patients. We also managed to source some homeless dogs and cats in animal shelters and bring them back to the university and conduct neutering surgeries on them to demonstrate that we could do surgery to the university's high standards. And we also were required to, as well as doing our own surgical course that we had to organise ourselves, we also had to attend the standard surgical program as observers. This was actually very beneficial because we got to compare the two types of surgical training. I also went and sourced my own DAISY surgical simulator, as I mentioned, and conducted some surgeries on that. And we also were able to conduct some abdominal and orthopedic surgeries on the cadavers of animals that were being sadly euthanized because of genuine medical reasons. So the outcomes of that was that we missed out as surgeon or assistant surgeon in 13 scheduled surgeries at most. There were 13 terminal surgeries in the curriculum, and each week the students would rotate, and there would be 3 students in a group. A student would be the surgeon or the assistant surgeon or the anaesthetist. So if there were 13 of those, you got to be the surgeon or the assistant in about 2/3 of those. But what we gained instead was performing or assisting with at least 62 additional surgeries that the rest of the students didn't get access to, performed under close one-to-one supervision in private practice. In the surgical labs, there would typically be about 6 surgical groups with 2 or 3 instructors. So there was not one-to-one supervision, but we did get one-to-one supervision in our externally arranged teaching program. Jointly, we ended up castrating or spaying 45 dogs and cats, including 21 spay operations, before we even entered the final year of our course. In comparison, the other students in our year, as was normal in veterinary schools at that time, got to do maybe 1 spay, maybe half a spay, maybe a few more, sometimes 0 spays before they graduated. And of course, the spay operation, the female ovarian hysterectomy, is a major surgery, and it's probably the most important surgery that new graduate veterinarians need to be able to competently perform. And it's a major source of stress for new graduate veterinarians because it is such a major surgery. We also participated in a wide range of other surgeries as well, and you can see those there. We had a similar depth and breadth of anaesthetic experience. And instead of simply doing a surgery, we were also exposed to the clinical presentation of the animals, the clinical histories, the preoperative examinations, the diagnoses, the surgeries, but also the postoperative management and recovery of all of those cases. So we got a much broader range of experience. Consequently, the very common problems for new graduates of competence and confidence were greatly alleviated in our case because we had so much more experience than most of our classmates had. This program was considered so popular, our animal shelter neutering program, that it was mainstreamed into the curriculum soon after that because we got so much more experience. And that sort of experience has been common at other veterinary schools around the world as well. An alternative program is often set up, often involving an animal shelter sterilisation program, which is then mainstreamed into the curriculum because it provides such a great wealth of experience for students. It provides such a useful community service, helping to allow these animal shelters to get these neutering operations done at low cost and increasing the adoptability of these dogs that then don't go on to contribute to the pet overpopulation problem the shelters are trying to solve in the first place. So, despite spending more time on my campaign for humane teaching methods than on my studies and consequently almost failing the veterinary course, I managed to scrape through in the end and went on to practice small animal practice, mostly around London for most of the next decade. I was then recruited to really because of I knew so much about clinical skills and surgical skills, training models and mannequins, to go and teach at Ross University School of Veterinary Medicine here on St Kitts. This was the temporary accommodation that I was placed into on arrival. And I had to deal with traffic passing back and forth in front of my windows, unwanted or unexpected, I wouldn't say unwanted, but unexpected visitors on a regular basis. And this was Ross University, the main campus. And this is one of the cafes at which the students and staff would eat their lunch overlooking the Caribbean Sea. So it was a very beautiful place. But students would often complain home on social media that they were not able to develop a tan because no one had any time to spend on the beaches, not very much anyway, because we got rid of the summer break and put an extra semester into the year. So there were 3 semesters every year, which meant that students could graduate early, but it was a real pressure cooker of an environment, both for students and for faculty. Everybody worked very, very hard. We had a clinical skills training laboratory, which we converted from a cafeteria. And because our student numbers were so high at Ross, we would run classes typically 6 times to fit everybody through these classes. We would start students learning basic suturing and manual skills in first semester of the curriculum and build in a stepwise fashion semester by semester. So by the time they came to start their actual surgical courses at the end of the curriculum, they were had a lot of experience and were relatively skilled and had a pretty good reputation when they went to do their final year externships at affiliated US mainland veterinary schools. For anyone that's interested in establishing a clinical skills laboratory, all the various logistical considerations, equipment considerations, staffing, and so on, with the person that actually established our clinical skills lab, we published a paper on how we did it and with guidance for others as well. I'll just draw people's attention to that. More recently, with some other colleagues, we surveyed EU member states to look at the amount of educational animal use that was occurring. We published a study in 2021 that described the years 2014 to 2018, and it showed that there was, apologies, looks like the bottom of the slide's been cut off, but there's between about 125 and 170,000 animals being used for educational purposes in the EU each year. Just before this presentation, I did a check for the latest numbers. Sadly, it hasn't fallen. It's still in that range. It appears that about 145,000 animals are being used for educational purposes in the EU each year. And I did a comparison with the US, and the estimates of US animal use are as high as 20 million animals per year, although I think that might be an overestimation of animal use for educational purposes. But nevertheless, that would be an amount that's more than 100 times larger than the educational animal use across all of the European Union. What's the difference in populations? Well, in the European Union, there's about 450 million people, and in the US, there are about 340 million people. So the US population is not 100 times larger than the EU. It's actually smaller than the EU. And yet we have educational animal use that could be 100 times larger occurring within the United States. And even if that is something of an overestimate, nevertheless, it indicates a massive disparity in educational animal use between the US and Europe. Educational animal use in the US appears to be vastly, vastly higher than in Europe. And that is clearly is a problem. We also surveyed the types of procedures that animals were being used for based upon the summaries of animal use that have to be provided in a publicly accessible form on the websites of the competent authorities in all EU member states that are responsible for overseeing scientific and educational animal use. There have to be publicly available summaries of this animal use. We found that, as you can see, the procedures in which animals were used most commonly were skills training, surgical training, and physiology. The animals used most commonly were rodents with some use of pigs and other species. The reasons why educators stated in all these summaries that they needed to use animals most commonly was because they felt that practice on a living animal is necessary for proper learning and that no adequate model or alternative was currently available. So we also did a systematic review of published studies that have compared learning outcomes when humane teaching methods are used compared to traditional harmful animal use. We searched 3 major bibliographic databases, ultimately finding 50 studies comparing learning outcomes, going as far back as 1968 up to 2020. There would be even more now. The humane teaching method produced superior outcomes for the studies indicated in blue and equivalent outcomes for those indicated in yellow. There's only a small number for which they produced inferior outcomes in red. The disciplines in which most of these studies occurred, and indeed in which most animal use occurred, was animal anatomy, physiology, and veterinary surgical skills training. And the humane teaching methods most commonly studied were computer simulations and models, for example, surgical training models. Overall, across all these 50 studies, the humane teaching methods produced learning outcomes that were superior in almost a third of these studies, equivalent in most of the rest of them, and inferior in only 10% of these studies. And they tended to be alternatives that were not well designed. The surgical skills assessed, as you can see, included psychomotor skills in all of these studies, abilities of students to perform ligations, intestinal procedures, gastrotomies, ovarian hysterectomies. And there are a range of other non-surgical skills that were assessed in these studies as well. Interestingly, when doing one of my earlier studies and publications in this field, I accidentally found 29 other studies of veterinary student outcomes which didn't involve comparisons with harmful animal use that studied these sorts of humane teaching methods. And I published a table describing all of these, the features of these studies in 2007. And common advantages of these humane teaching methods were these: time and cost savings, often very substantial. And indeed, this is the biggest reason that seems to have driven the widespread adoption of these humane teaching methods in life and health sciences education across the globe, of which veterinary education has been lagging the most. Repeatability and flexibility of use. Students have the ability to repeat procedures quickly and easily, unlike dissecting an actual animal. Once it's done, or once the experiment's been conducted on an animal and the animal's been killed, then it's not easy to quickly repeat it. Customization of the learning experiences to student learners' individual needs. They can often do that. Of course, there can be savings of substantial number of animal lives and increased compliance with legislation and coded practice about using alternatives. One point that people often don't think about is decreased student exposure to toxic chemicals used to preserve dissection specimens. There have been studies showing that aerosolized levels of formaldehyde in dissecting rooms often exceeds allowable safety limits. It is possible that students that might have an adverse reaction to some of these chemicals, which by the way are often carcinogens, could potentially sue an educational institution if those limits are breached. And that actually often is the case when you look at studies of the air quality in those rooms. And of course, there is decreased potential for conflict with students unwilling to harm animals during their education, such as myself and other students around the world that have also generated adverse media coverage for universities and sued their educational institutions, usually successfully. There's another thing as well, the actual learning experience of students. This is a survey of 370 vet students doing first year physiology at the University of Illinois in 1999 by Linnea Stoll, who was a colleague of mine. She was also a student at the time. In first year physiology, more than 100 dogs, pigs, rats, and rabbits were killed to demonstrate well-established physiological principles. And the students were surveyed by Dr. Stoll about how they felt the experiences had been for their learning. And I'll let you just read some of those statements there for a minute. And some more on the next page. I don't know about you, but certainly this speaks to our experience in our physiology laboratory class. The instructions about how to actually anaesthetize all of the sheep that were being anaesthetized by second year student groups were very patchy. Students hadn't prior experience at these procedures, and there might have been 2 or 3 demonstrators for a class of 30 with maybe half a dozen or more student groups and sheep on tables. Accordingly, the level of anaesthetic maintenance was often inadequate. Some sheep became too light and presumably were able to experience pain and suffering. And some sheep went too deep and were accidentally killed by students prior to the laboratories being ended. And this was not uncommon. This would be the norm, I think, in these sorts of laboratories where students are highly inexperienced and the supervision and instructions are grossly inadequate. You can see the overall results. About 60% believe these animal labs were, quote, not worth the resources used, and only 20% that felt they gained great benefit in their understanding of physiology from the laboratories. There was a similar study of veterinary students at Massey University in New Zealand around about the same time. And there was a dramatic drop in how useful students felt that these sorts of laboratories were once they reached the final clinical year of their course and they gained familiarity with what vets really needed to know to be successful in clinics. That enabled them to put the value of these first year laboratories into much greater context. And their support for these laboratories in New Zealand crashed precipitously once they got into doing their clinical experiences. Because of that survey, most of those labs were stopped at Massey, and because of this survey here, it put the labs under great pressure at the University of Illinois, but they didn't stop until I advised Linnea Stoll to go to the media. And the adverse publicity and pressure upon the university that resulted caused more progress in 10 days than the previous 10 years, and these labs were then stopped at the University of Illinois. I want to, in this closing part of my presentation, note something else that's going on as well, another adverse impact of this harmful animal use. If you think about it, we have the dissection often of purpose-killed animals or those from unethical sources within anatomy. We have demonstration experiments demonstrating scientific concepts that have been well established for years or decades in physiology, biochemistry, and pharmacology. Animals are also used as disposable teaching tools in these experiments. And then we have animals being killed in surgical laboratories. And yet the majority of veterinary students receive little formal education in animal welfare issues, critical reasoning, or indeed animal ethics. At the same time, they're being directly required to harm and kill animals during their education. And the usually unspoken messages are that harming and killing animals is not only condoned, but required to become a vet. You can't become a good surgeon unless you do this. And animal welfare concerns are subservient to human interests of very debatable merit. So this is giving negative underlying messages about the intrinsic value of animal lives. They're being seen as disposable teaching tools. And it results in a great deal of stress to students who have come into the course in order to learn how to become healers of animals. And probably for the first time in their lives, for most of them, are being asked to actually seriously harm and kill healthy animals. We know from studies of learning that stress, severe stress, impairs learning ability, impairs cognitive processes, memory, recall. So it actually decreases the efficiency of the learning process. We have seen in studies of veterinary students that this can result in desensitization as well, including diminished capacity for compassion and ethical decision making. There have actually been a series of studies demonstrating these sorts of phenomena, specifically the decreasing awareness of veterinary students of the hunger, pain, fear, and boredom of dogs, cats, and cows as their veterinary courses progress, decreased likelihood of final year veterinary students to provide appropriate perioperative analgesia when compared to junior veterinary students, an inhibition of something called moral reasoning ability, which developed in all other student cohorts, but not veterinary students during the 4 years of veterinary school. I would note that there have been more recent studies actually also demonstrating these phenomena. So these are actually, I've said in my writings, these are actually desensitization related phenomena. They're actually psychological adaptations. They arise from cognitive dissonance. On the one hand, students they believe initially that animals are sentient and can suffer and be hurt by these sorts of procedures, and yet they're being required to participate in these procedures and cause animals to be harmed and killed. As human beings, we are motivated to resolve discord created by cognitive dissonance. And we've got two ways we can do that. One is that we can change our beliefs, and the other is that we can change our behavior. Changing the behavior for the veterinary students whilst maintaining those beliefs would mean not participating in these procedures. That has historically been very high risk for veterinary students. When all the messages are that you must participate in these procedures or you won't become a competent surgeon, and indeed if you boycott these laboratories, you might well be failed out of the veterinary course. And this is very high stakes for students that may have up to $250,000 in debt, very high student debt levels in the US. And if the students are failed after having worked incredibly hard to get into these programs, if they fail, they won't have any conceivable way to actually pay that money back. So the pressure on students not to change their behavior, to continue to participate in these programs and not risk being failed out of these programs is enormous. So what most students do is they don't change their behavior, instead they change their beliefs. And that means convincing oneself that maybe the animals aren't suffering quite that much after all. Maybe they're not as sentient as we initially believed. Essentially, desensitizing over time. So these are psychological adaptations that enable previously caring students to withstand psychological stresses resulting from these requirements to harm and kill sentient animals. So I have, in another study, documented the impacts that these desensitizations have upon practicing veterinarians, and in particular their professional associations. It essentially means that the positions of professional veterinary associations around the world and internationally on animal welfare and animal use issues often lag behind those of the general public. So instead of leading on issues of animal welfare and animal ethics, which the general public quite rightly expects veterinarians to do, vets, when you do the surveys, it appears that they actually lag behind those of the general public on animal welfare and ethical issues. That's completely unacceptable. And it results because of, I think, because of these desensitization related adaptations that students have been forced to undergo as a way to survive these intolerable requirements to harm and kill sentient animals during veterinary school. So accordingly, as well as directly saving the substantial number of animals that are killed within veterinary and other curricula, humane teaching methods enable students to graduate without needing to go through this kind of desensitization and loss of their ethics. Many of the graduates have higher animal welfare and ethical standards, which of course is more likely to result in benefits directly to future patients when vets more accurately understand that the animals in front of them are sentient, are likely to suffer, do warrant appropriate perioperative analgesia, and more likely to result in veterinary professional associations that actually do lead rather than follow evolving social standards on animal welfare. So, to conclude, well-designed humane teaching methods usually perform at least as well as methods relying on harmful animal use, in some cases achieving superior learning outcomes. It's obvious then that educators can best serve their students and animals whilst minimising financial and time burdens upon their faculties, which are increasingly important today, by the use of well-designed humane teaching methods. For anyone that would like advice, such as for students seeking to conscientiously object to harmful animal use within their courses, and this is something that goes on all the time. I've been in touch with students earlier, well, a student earlier this week at an Australian veterinary school going through this. Students go through this at veterinary schools around the world on a regular basis. there is advice and support available for them, along with alternatives libraries where people can actually borrow alternatives for free to see what they're like, and access to free online computer simulations and so on. You can find them on these websites here, humanelearning.info, that's mine, and also the Interniche site, that's the International Network of Individuals and Campaigns for Humane Education, which was mostly veterinary students going through these experiences at vet schools around the world of asking for alternatives and having to deal with the opposition of their universities usually. And mostly now they're all graduates and have usually been very successful in veterinary practice, as was I, despite using alternatives in our training programs. I'd also direct everyone to Barry Kickman's excellent recent book on veterinary ethics. You've got many great chapters in there, and kindly asked myself and one of my co-authors to publish a chapter on animal use in veterinary education. So we've got more recent information there about animal use in US veterinary schools, and we summarise a lot of the information on alternatives that I've just provided you with. Again, that's my website, humanelearning.info. If anyone would like to find advice and support, links to free online alternatives and so on, you can find it there. And then I'm happy to take questions if anyone has any of those. Thank you very much for joining.
That's fantastic. Thank you. And there we go. You want to stop sharing so we can, yeah, anybody who would like to stay, I'm going to stop the recording, however, just so that people can speak freely. That way, if I find it, I'll figure it out here in a second. But I do have one question before everybody else, before you take one. Can you talk about ethically sourced cadavers like Dr. Dixon's? Where can you give us an example of that?
Sure. So Tufts University, for example, in the US, at least